Hello everyone, thank you all for joining me today. I would like to give a little talk about our research regarding the effects of various factors that affect de novo genome assemblies. Our research objectives were divided into two parts. First, we want to find out the effect of sequencing coverage and input library on the quality of de novo assembly. Our hypothesis for this part is that a higher coverage will lead to a higher quality assembly. Also, we hypothesize that using a synthetic long-read library will produce a higher quality assembly. For the second part, we want to look at the effect of genetic heterozygosity. Our hypothesis for this part is that a higher ploidy and more somatic variants will both negatively affect the quality. For our organism of interest, we chose Xylocopa virginica due to the unique method of sex determination, a process called haplodiploidy. Throughout this process, an unfertilized egg will develop into a haploid male, whereas a fertilized egg will develop into a diploid female. We obtained 4 samples and sequenced them using Illumina and assembled using Supernova 2. We have a young male, a young female, an old male, and an old female. We also have 1 diploid young female sample sequenced using 10x linked reads and assembled using Supernova. Now let's take a look at some results. For the 4 Illumina parent libraries, between the coverages of 5 times to 40 times, we find that the diploid samples both show N50 value peaks at 20 times coverage. In comparison, for the haploid samples, the assembly quality actually improves with the increasing coverage, with the unworn male or the young male sample showing the most improvement. On the other hand, for the 10x linked reads library, the quality peaks at a 63 times coverage and then decreases afterwards. So when comparing between the 10x assembly and the Illumina assembly, the 10x assembly actually has a 22 times higher N50 value than that of the Illumina assembly. Let us now take a closer look at the 4 Illumina assemblies. The overall ranking of the qualities start with the unworn male as having the highest quality, followed by the worn male, the unworn female, and finally the worn female. By comparing between the N50 values, we find that the haploid samples offer better genome assemblies than the corresponding diploid samples. The unworn male versus the unworn female show that the unworn male has a 100 times better N50 value, whereas the worn male has a 45 times better N50 value than the worn female. When comparing between the different age groups between males and females, we find that the younger samples offer better assemblies than the older samples. We also notice that the males have a larger degree of difference than the females. This is most likely due to the fact that diploid females are able to take advantage of homologous chromosomes as a DNA repair mechanism, and thus less somatic variants are accumulated. Through our research, we discovered that there is a noticeable trend between the samples. When looking at the effect of sequence coverage, we see that the young male sample had the biggest improvement in N50 value. In addition, the unworn male sample had the highest overall quality. In fact, the N50 value for the unworn male was more than 100 times that of the lowest ranking assembly, the worn female. We conclude that ploidy, the age of sample, sequence depth, and sequence library all have variable effects on the assembly quality. For future de novo genome assemblies, we recommend the use of haploid samples at the youngest possible age. We also recommend sequencing at a high coverage using preferably a long-read platform. And so with that, I'd like to acknowledge the people who have contributed to the work as described here. I'd like to thank my mentors and collaborators, Dr. Liang, Dr. Skandalis, and Dr. Richards, as well as my lab mates from the Brock Liang Lab and Lynn Duff from the Brock B Lab.